Hey everybody, welcome to the very first lecture of Biology 6. This is generally the first chapter of any biology textbook that you use. For example, the Campbell text, which is shown here, chapter 1 is called The Introduction, the Themes of the Study of Life. But if you look at generally any biology textbook, you'll find that usually the first chapter deals with some of the major themes when we study life on Earth. So, welcome to the course. You're going to hopefully have a lot of fun, and let's get started.
If you had to kind of guess at what the overall theme of biology is based on, my answer to you would be evolution. Everything we study in biology is with that sort of framework of evolution, the process of change that has transformed life on Earth. For example, an organism's adaptation to its environment and how it functions in its environment, its physical appearance, its biochemical activities, are all the result of evolution. This mother of pearls plants thick, succulent leaves, or an adaptation to conserving water. This helps it survive in the crevices of rock walls. What happened to enable this plant to conserve water occurred basically as a result of environmental pressures, and it was reflected in changes in its genetics and changes in its physiological ability. So its biological function started with a change to evolutionary pressures. Of course, many questions remain about these changes, and asking these questions and finding the science-based answers is what we call biology.
Biology is the scientific study of life. Biologists ask questions that start with the word how. They ask questions that start with the word why. So you see here a picture of two male seahorses showing camouflage so that they can blend in with their environment. A biologist might ask the question of how does this seahorse camouflage itself? Other questions biologists could ask could be how does a single cell develop into a multicellular organism? How does the human mind work? Even a question like how do living things interact in what is known as a community? These questions are really important. The problem is the answers are not so simple. As a matter of fact, if you looked at life, life defies a simple one-sentence definition. So the question is, what is life? What are the characteristics of life? This slide gives you the 10 characteristics that when you put them all together, say you have a living thing. This organism must be capable of movement, both internally and externally. It must be responsive and be able to adapt to its environment. It must be able to grow. It must be able to reproduce. It must be able to digest nutrients and process those nutrients into energy. It must be capable of absorbing things. It must be capable of secreting things. It must be capable of circulating things within its cell and even within the multicellular organism. It must show order in its structure. And finally, it must be able to regulate.
This slide lists the 5 requirements for life on Earth. They're relatively straightforward, and you probably know 4 of them already: water, food, oxygen, and heat. However, the 5th is called pressure. Now, this does not mean, oh my God, I have an exam tomorrow and I haven't studied for it. This actually refers to atmospheric pressure.
Biology is more than just memorizing factual details. Yes, it is true in biology that we have to memorize things. As a matter of fact, the process of learning starts with memorizing. And then what you have memorized eventually becomes short-term and then long-term memory. And that long-term memory we call learning. So yes, there is some memorizing factual details involved in biology. But what also helps is to take those factual details and organize them into themes. So these themes that we create in biology help organize that biological factual information that we are trying to learn. The first theme is new properties emerge at each level in what is known as a biological hierarchy. Think of a biological hierarchy as a staircase. The bottom step, the next step, the third step, etc., etc. And you can use this biological hierarchy to study life at different levels, from things like atoms and molecules to an entire living planet. So we study life by dividing them into this hierarchy, and this hierarchy is made up of different levels of biological organization. The bottom step of that staircase would be the atomic level. The top of that multi-step staircase we would call the biosphere. And as we go from step to step to step, different properties of what we are studying at that particular step emerge. And these properties are known as emergent properties. The one thing to realize about emergent properties is that they are unique at each step along the biological hierarchy. This is because they result from the arrangement and interaction of the parts within that system, within that level of hierarchy. For example, the human brain, memories and thoughts are emergent properties, and they have emerged because of the complex interaction between neurons. Or photosynthesis in chloroplasts. That's an emergent property because of the interactions of the parts of a chloroplast. Photosynthesis will not take place in a test tube if you just toss a bunch of disorganized chlorophyll parts in there. They have to interact and arrange themselves in a very specific way in order for the emergent property of photosynthesis to appear. That's what this picture is trying to show you. It's trying to show you the biological hierarchy. Starting off down in the bottom right-hand corner here, we have atoms, which combine to form molecules. Those molecules will form the subcellular organelles of a eukaryotic cell, or those molecules can combine then to form actual cells if they don't have organelles. The cells combine to form tissues. The tissues become organs and organ systems. We then move up from organ systems to organisms, which are individual living things. Take a bunch of individual living things living within a certain area, a certain geographical area, like the population of sugar trees, maple trees within a forest, and you have a population. Put a bunch of populations together in a geographical area, what is known as an ecosystem, you got a community. And take those communities and combine them together in an ecosystem, together with all of the non-living components of that ecosystem. And that's what you end up with. And then take all the ecosystems on the planet and you end up with the biosphere. Each of these categories you see on this chart, each of them have a series of unique properties that emerge as we go from level to level to level. Now, this doesn't mean that the biosphere doesn't have the emergent properties of atoms. You do not lose these emergent properties as you go up the staircase. So molecules will have a new set of emergent properties in addition to the atomic emergent properties. Cells will have a new set of emergent properties, but they don't lose the molecular properties or the atomic ones, and so on, and so on, and so on. Emergent properties are not unique to living things. They can characterize non-biological entities as well. For example, a functioning bicycle emerges when all of the necessary parts connect to one another in a very specific and correct way. Or here, down here in this picture, we have the structure of diamond, the carbon arrangement in diamonds, and the carbon arrangements of graphite. And the graphite lead of a pencil and a diamond are both built of carbon molecules, but the way they arrange their carbon atoms are different, leading to very, very different emergent properties. A lead pencil is not a diamond. To be honest, as we climb up through the biological hierarchy, these emergent properties become more numerous and more complex. And that does make it very difficult to study them as we climb up that biological hierarchical staircase. So as biologists, we try to reduce them into something simpler. This is known as reductionism. Reductionism is the reduction of complex systems to simpler components that are more manageable to study. So you would study one aspect of that biological hierarchy, and then as you begin to learn about that particular component, learn about another component, learn about another component, and then try and tie those components together. Again, biology is not just about memorizing facts, it's about tying these things together. So in reductionism, we take a complex system, we break it down into its constituent parts, we study each part, and then we try and assemble that together as a big picture. You need to observe the interactions of these component parts if you want to fully understand the system. For example, studying the molecular structure of DNA helps us to eventually understand the chemical basis of genetic inheritance. But you need to study how DNA Might interact, for example, with the proteins of the nuclear matrix so that it forms chromatin. And then you might need to understand how DNA gets replicated so that it can be passed on to daughter cells. Then you might need to understand how mitosis takes that replicated DNA and divides it into daughter cells. And if you're a geneticist, you might even go even further to understand how a particular gene on a section of DNA gets expressed in a daughter cell and changes its phenotype. So you can see the complexity of biology. And we try not to get overwhelmed with this complexity by using reductionism. I have a phrase, KISS, KISS, keep it simple, stupid. And I swear to God, if you keep things simple in biology at the start, it will allow you to build up and become more and more complex in your understanding. So keep it simple. But once you've kept it simple and you understand the way these simple organizational components work with each other, you do have to put it together into a larger, more holistic picture, what I call the big picture. So for example, how cells and interact with one another to become an organism, how organisms interact within a population, or how populations work together to form a community. This scaling up of reductionism to larger pictures is called systems biology. And system is a combination of components that function together. It's like you study how each of the individual components of a bicycle work in reductionism. And then systems biology is how you understand how those parts get put together to make a bicycle. Systems biology constructs models for the dynamic or changing behavior of an entire biological system. Systems biology is based on studying the interactions among the system's parts. And the important thing about systems biology is if you can understand how each individual part works, and if you can figure out how those parts work together, then you can start to make predictions when one variable changes. If you change one aspect of one part, how is that going to change how the system works? If you suddenly put in a new, fancier gear system in your bicycle, how does the bicycle change its function? Systems biology is important in things like pharmacology, physiology, in medicine. For example, how does a drug for blood pressure affect the other organs of that organism? That would be a physiological systems biology question. You can scale it up to the entire planet. You can use systems biology to study ecosystems. How does increasing carbon dioxide alter the biosphere? Theme number 2 tells us that organisms interact with other organisms, and they also interact with their physical environment. In an ecosystem like the one you might see here, this elephant eating a tree, and you can see we're on the savanna in Africa. In that ecosystem, every organism interacts with its surrounding neighbors. It also interacts with the environment. And this environment includes non-living factors, in addition to the living other organisms. So here we would see the leaves of a tree taking up carbon dioxide, producing energy through photosynthesis, and making oxygen as a byproduct. The oxygen is released into the air for the other organisms to breathe and use to keep themselves alive. Like us, we have an oxygen, we are oxygen breathers. It is the plants on the planet undergoing photosynthesis that have created our oxygen environment. This elephant right here is eating the leaves of this tree because photosynthesis produces sugar. It is extracting the nutrients from the sugar in that leaf. It processes that energy and uses it to keep it alive. And then the unprocessed food goes out the other end. And into the soil, that waste will then become incorporated. The bacteria in that soil might use some of the nutrients of the poop. Heck, there's bacteria in the poop themselves. Those bacteria enter into the soil. The soil gets that much more healthy and nutrient rich, makes the tree grow bigger. Now, both organisms and their environments are affected by interactions, and most of these interactions are very, very good for each other, or good for one another. But let's face it, some interactions can be harmful. For example, greenhouse gases. As we begin to rip down more of these trees, there's less carbon dioxide that organisms can extract out of the atmosphere. And suddenly, instead of being 0.04% of the environment, carbon dioxide increases to 0.06%. And that small change acts as an insulating blanket to prevent heat from escaping from our environment. And suddenly our planet warms. That is an example of a harmful interaction. Theme number 3 is that life requires energy transfer and transformation. A fundamental characteristic of all living organisms is the fact that they can use energy to carry out their activities. They can extract energy from food and use it to move, to grow, to reproduce, etc., etc. But living organisms don't just take one form of energy and use it. They have the ability to transform one form of energy into another. For example, in plants, they have the ability to take light energy and convert it into a chemical energy known as sugar. This is the process called photosynthesis. That chemical energy can then be broken down into kinetic energy. Cells can take that sugar and use it to make ATP. And that ATP can be used to drive the movement of muscles. So that ATP is used to contract muscles so that the organism can move. That ATP can be used to drive reproduction, to drive anything we had on that previous slide, circulation, secretion. You get the idea. But that chemical energy, which came in the form of a sugar, was converted into ATP, which then was converted into a kinetic energy like growth or movement. The energy flows through an ecosystem, usually entering as light and exiting as heat. Before we leave this slide, though, there's one very important thing you need to realize about energy. You can't create it, you can't destroy it, you can only transform it from one form to another. That transformation is not 100%. For example, the energy entering into a leaf as light energy does not become 100% transformed into chemical energy sugar. Some of it is lost to the environment as a form of energy known as heat. And all living organisms on the planet will do this. They will take in one form of energy, convert it into another one, and because that conversion is not 100%, and because you can't destroy energy, that energy is usually lost in the form of heat. Theme number 4 says that structure and function are correlated to one another at all levels of that biological hierarchy. One way to think about it is that form fits function. So if you analyze a biological structure, you can pretty much figure out what it does. For example, take a look at the bones here of this bird's wings. They are designed to be strong. That's what these little bony struts going across are for. They give the bone strength. But notice all of those little air spaces. It makes the bone strong and light. And this allows the bird to have strength, but yet have enough lightness to lift itself off the ground when it flies. The structure of an elbow joint tells us that it functions like a hinge for flexion and extension. The structure of a chloroplast can give us an idea as to how it functions in photosynthesis. The fact that leaves are thin and flat, they function as light capturing devices. So form fits function. Structure and function are correlated to one another across all steps of that biological hierarchy. Theme number 5 is the cell is an organism's basic unit of structure and function. In other words, the cell is the lowest level of organization that can perform all activities required for life. Cells can move, cells can reproduce, cells can grow, cells can circulate, cells can absorb, cells can secrete, cells have order, etc., etc. That doesn't mean that the cell is the lowest level of organization on our biological hierarchy. No, that's the atom. But the atom does not have the emergent properties, those 11 or 12 emergent properties that characterize life. It's only when you get to the cell that you will see all of the activities required for life. Now, all cells, be them prokaryotic or eukaryotic, are enclosed by a plasma membrane, and all cells use DNA as their genetic information. We are going to study prokaryotic cells in this course a little bit. If you've taken microbiology, you have studied prokaryotic cells a lot more. We're going to spend most of our time in this course, however, learning about the eukaryotic cell. And we'll see the differences between these two kinds of cells. For example, a eukaryotic cell has membrane-bound organelles, the largest of which is a nucleus. In comparison, prokaryotic cells, they do not have membrane-bound organelles. They do not have nuclei. They're also significantly much more simpler and smaller than a eukaryotic cell. But I don't care about whether you're a prokaryotic or a eukaryotic cell. Both of those cells are surrounded by a plasma membrane, and both of them use DNA as their genetic information. And both of them are capable of performing all activities that are required for life. Theme number 6 is that the continuity of life is based on the fact that DNA is passed on from one offspring to the next offspring to the next offspring. The continuity of life is because we pass on DNA from us to our children. So the ability of cells to divide and pass on that genetic information to the progeny, daughter cells, that's the basis of life, or I should say the continuity of life. It is the basis for the growth and repair of multicellular organisms. The dividing cell is going to pass on the genetic material known as DNA. This DNA is usually in the form of something known as a chromosome. Chromosomes do contain most of a cell's genetic information. That's going to be in the form of DNA, or deoxyribonucleic acid. DNA is the substance of genes, and genes are the unit of inheritance that are going to transmit information from parental cells to daughter cells, or more holistically, from parents to their offspring. We're going to spend a lot of time talking about DNA in this course. We're going to do a lot of molecular biology in Biology 6. Theme number 7 is that feedback mechanisms are used by organisms to regulate their biological systems. Now, if you've taken physiology, you've probably spent some time looking at these feedback mechanisms. You may have taken feedback mechanisms in other courses like Biology 3. Feedback mechanisms allow biological processes to self-regulate. There are two feedback mechanisms. There's negative feedback and there's positive feedback. In negative feedback, you have the accumulation of a product, and that product basically feeds back to affect one of the steps in this biological metabolic pathway. So for example, here we have product D. Product D accumulates and feeds back to affect enzyme 1. It may bind to enzyme 1 and shut it off. The resulting steps in this biological metabolic pathway are going to stop, so that the level of D begins to decrease. Now, the opposite to a negative feedback system, which is what this is doing, this is shutting off a pathway. The opposite to negative feedback is not positive feedback. The opposite to negative feedback is the feedback is gone. As the amount of D produced in the cell decreases, the negative feedback is basically removed. Enzyme 1 turns itself back on, begins to convert product A into product B. B becomes C, C becomes D, and that metabolic pathway picks up again. Usually systems, negative feedback systems, will allow for a particular amount of a product to accumulate. When it gets over that amount, we then see that negative feedback. When it drops below a specific amount, the negative feedback mechanism is removed. So positive feedback is not the opposite to negative feedback. If you shut off negative feedback, there is no feedback. If positive feedback, this means that as more of a product is made, for example, Z right here, it actually again feeds back to one step in this biological pathway, and it actually promotes that step. It speeds up that step so that more of that product is produced, more feedback is generated, more of that product is produced, etc., etc. The opposite to positive feedback is to remove the feedback. Positive feedback mechanisms are not as often, you don't see them as often in cells because positive feedback mechanisms can be very dangerous. Kind of like a speeding car getting out of control. There are some positive feedback mechanisms that are used. For example, there is one used by the female reproductive system to stimulate ovulation. But by and large, most feedback mechanisms you will see in biology will be the negative feedback mechanisms. Okay, we've looked at 7 themes. I'm about to give you the biggest theme of them all. This is the granddaddy theme, the theme to beat all themes, and it is evolution. There is consensus amongst biologists as to the core theme of biology, and that's evolution. Theodosius Dobzhansky said, "Nothing in biology makes sense except in the light of evolution." This is because evolution unifies biology at different scales of size throughout the Earth and throughout the history of life on Earth. It makes sense of everything we know about biology. Evolution allows us to explain why life is so diverse on this planet. It also allows us to explain why many diverse species can share characteristics with one another. Did you know that human beings share 98% of their DNA with chimpanzees? Evolution explains that, because at some point in the evolutionary history of chimpanzees and humans, we shared a common ancestor. All organisms on this planet are modified descendants of common ancestors. And as that descending took place, the descendants became modified. They adapted to their environment. They descended from their common ancestor. They adapted to the changing environment. And those adaptations were then shall we say, precipitated or became evident in the characteristics of the organism. So if you can't descend from a common ancestor and adapt to a changing environment, you go extinct. When you descend from a common ancestor and adapt to the changing environment, your species survives. And it explains why while descendants might look different from one another, but they share characteristics with one another because, well, those characteristics existed in the common ancestor. Now, there may be some of you in this course that have been raised in religious households, and you may have certain religious beliefs. And I think that is spectacular. I wish we were in the classroom and I wish we could be seeing one another and having a great conversation. It is difficult when you try and marry the concepts of evolution with the concepts of religion. Let's face it, religion and science, they kind of rub against each other sometimes quite painfully. I want you guys to know that I respect those thoughts and I respect that education and I respect those opinions. However, this is a class, this is a scientific class. So I will not give religious explanations for anything. I will always give scientifically proven explanations for the concepts that we will discuss. Life is incredibly diverse. Approximately 1.8 million species have been identified and named to date. Thousands more are being identified every year. There are over 100,000 species of fungus, 290,000 plant species, close to 52,000 vertebrate species. These numbers are climbing. Over 1 million insect species have been identified, and untold numbers of single-celled species. There are estimates that the number of species on this planet could be anywhere from 10 million to 100 million. Thousands of new species are identified every year. Of course, thousands of old species go extinct every year. But evolution can allow us to look at these species and find commonalities among many of them, because every single one of those 100,000 species of fungus descended from a single common ancestor. Every single one of the 52,000 vertebrate species on this planet descended from a common vertebrate ancestor. So that's how evolution becomes the overarching theme for biology.
Taxonomy is a branch of biology that names and classifies species into groups of increasing breadth. The biggest group would be called domains, and after that you would end up with a kingdom, and then kingdoms would be made up of several phylums, and a phylum would be made up of several classes, etc., etc., etc. So over here on the right hand side, this would be the biggest taxonomic class, and as we go to the left, they become more and more narrow and more defined in their scope. To be honest with you, we don't really use the word kingdom in taxonomy, but it still hangs on because it's a really nice way of classifying things. So don't be surprised if you move on to, say, a course like Biology 7, where you will learn that the kingdom branch no longer exists, or the kingdom group no longer exists, but yet you'll hear me use it in Biology 6.
Now, back in 1969, Robert Whittaker was attributed to creating what is called the five kingdom system. We did recognize that there were two fundamental cell types. There were prokaryotes and eukaryotes. The prokaryotes went into what is called kingdom Monera, and the eukaryotes went into the remaining four kingdoms: Protista, fungi, plantae, and animalia. This classification scheme was based on nutritional requirements. It was based on how they acquired their nutrients. It was based on also physical characteristics. And I can tell you that once DNA sequencing got to be a lot easier and cheaper, this five kingdom classification system got thrown out the window. When I was in university in we had the five kingdom classification system. When I started teaching in about the year 2000, we had a 3-domain system. And how we went from a 5-kingdom system to a 3-domain system was because the molecular biologists were allowed to ruin everything. Well, we were allowed to give our input based on sequencing DNA. Caused a little bit of reorganization. For example, it took the kingdom Monera and split it up into 2 domains, one called Archaea and one called Bacteria. And it allowed for the creation of a 3rd domain of the eukaryotic cells, which is called Eukarya, or you can call it Eukaryota. So because of this molecular data, we went from 5 kingdoms to 3 domains. The remaining 4 kingdoms, Protista, fungi, plantae, animalia, they got shoved into domain Eukarya. It is thought, though, that those 3 domains share a single common cellular ancestor. Now, we don't know what that cellular ancestor is. I don't think anybody has found evidence of it, but it is thought that there is a common ancestor, or maybe 1 or 2 common ancestors, that gives rise to all life on this planet. That gives rise to the diversity of life, but it also explains the striking unity that we see. Here we see a cilia on the outside of a paramecium. This is a kind of like a small hair-like structure, which is found on the outside of paramecium, and it allows the paramecium basically to swim around in its freshwater environment, to trap food and to eat and to reproduce, etc., etc. What you see is a cross-section of the microtubules that are found making up that cilia. It is the exact same cross-sectional organization of the cilia found in the cells lining your trachea. Now, nobody would say, hey, we are descended from a cilia. I don't know, maybe we were. Maybe 3 billion years ago, our cells and the cells of the cilia came from the same common ancestor. Why? Because the cilia has the exact same structure. Another reason for this unity is DNA. It is the universal genetic language common to all organisms. So to put it very simply, how do you account for the unity and diversity that we find in nature? Biology tells us it's evolution. Okay, this is just a funny picture. Go ahead, laugh, but I guarantee you, you're going to be the guy on the right-hand side all for the next, 18 weeks. I know I'm developing a permanent stoop. So who was the biologist that came up with this groundbreaking idea of evolution? Well, there are several people that began to work on evolution. One of them was Aristotle, because Aristotle was a smarty pants. He knew everything. But most people attribute the theory of evolution, or what I like to call the science of evolution, to this gentleman right here, Charles Darwin. Over 150 years ago, Charles Darwin developed this scientific explanation for the diversity and unity of life. This is because at the time he was graduating from school, scientists had made three key observations about life. They had discovered that organisms were suited for life in their environment. Organisms had very specific adaptations that made them reproductively successful in their environment. Their children went on to become reproductively successful in their environment. They also found that many forms of life share characteristics, but they also recognized that many forms of life were very diverse. Charles Darwin took all three of these key observations and formulated what he called the theory of evolution, which he called descent with modification. So when Charles Darwin graduated, he signed on to this ship you see here, the HMS Beagle, His Majesty's ship Beagle, as what was called a naturalist. This is somebody who was a member of the crew of the HMS Beagle. The HMS Beagle was doing a trip circumnavigating in the southern hemisphere. Specifically, they were sailing around South America, and then they were going to kind of tootle around the southern part of the hemisphere of the planet. His job was to observe living organisms, what we call flora and fauna, that would be both plants and animals, and categorize them based on his observations. He collected many samples. He was particularly fascinated with birds. And so he spent several years on the HMS Beagle observing the nature that was around him. And from that time, as a naturalist on the HMS Beagle, he wrote in 1859, The Origin of Species by Means of Natural Selection. Now, I can tell you, I took a course in, third year university, or maybe it was grad school. I can't remember. But our professor assigned us to read The Origin of Species. I'm going to be honest with you. I made it to the end of chapter 1 and thought if I have to read the rest of this book, I'm going to kill myself. So I'm going to tell you, if you ever suffer from insomnia and you need something to put you to sleep, read The Origin of Species by Means of Natural Selection. But I will tell you something interesting about this book. Never, ever, ever did Charles Darwin call his theory evolution. He called his theory descent with modification. And that's really what evolution is. It is descent from a common ancestor with modifications. Going back to the chimpanzee human, we had a common ancestor in our evolutionary history. The ancestor then diversified into two organisms. And let's say these two organisms, one went, one direction, the other went off the other direction. So that these two organisms ended up in two very, very distinct environments. Now I'm going to tell you, your goal as a human being is not to be a parent. Your goal as a human being is to be a grandparent. Your job on this planet is to have babies that grow up to have babies of their own. So we have these two organisms, one going off to one environment, the other going off to another environment. Two different environments, but both of those organisms have to survive to reproduce, and their offspring have to survive and reproduce, etc., etc. So they began to physically and biochemically and physiologically adapt to their environment as that species began to reproduce. It is the offspring that are capable of adapting to the environment, surviving and having offspring of their own. Those are the ones that survive. And if your modifications allow you to survive your environment and have reproductively healthy children, you win. If your modifications actually prevent you from reproducing successfully, eventually you and your offspring, you and your species will become extinct. That's what happened with the chimpanzee, and that's what happened with the human being. They migrated into two distinct environments, or even if they were in the same environment, genetic changes changed their offspring and changed their success at reproduction. Now, both of them are evolutionarily successful species. We have chimpanzees and we have human beings, right? But both of them were able to have offspring that were capable of responding to the environment through genetic modifications that allowed them to be better reproducers. We can now define evolution as a change in the genetic composition of a population from generation to generation. It is the fact that mutations happen, they change genes. If those genes are expressed and allow you to be a better reproducer, that's a great mutation, and that is going to drive evolution. If these genetic changes, these mutations, make you a poor reproducer and your offspring do not survive, that's a bad genetic change. That's a bad mutation, and that is going to cause your species to go extinct. The pattern of evolutionary change is revealed by data taken from a wide range of scientific backgrounds. Biology is involved in evolution. We have to consider geology, physics, chemistry. All of these areas work together in the overarching scientific field known as evolution. Now, Darwin proposed that similarities between organisms was again due to descent from a common ancestor in the remote past. These descendants, living in various environments, or what you could call habitats, responded to the environment by developing adaptations that allowed them to fit their habitat and allowed them to have reproductively successful offspring. He called the mechanism of this evolutionary adaptation natural selection. Now, you have heard people say natural selection is survival of the fittest, and that is true, because a fit organism has reproductively successful children. The more reproductively successful you are, the fitter you are. So nature will select for those reproductively successful individuals. That's natural selection. Here are 3 orchids. They are all orchids, but look at the differences amongst them, but yet they all have the similar kind of organizational pattern. This is because all 3 of these orchids descended from a common orchid ancestor in the remote past. Okay, here are a few things to know about natural selection. Natural selection is a process in which individuals with certain heritable traits survive and reproduce at a higher rate than other individuals who don't have those traits. Over time, natural selection can increase the match between an organism and its surrounding environment. And number 3, if an environment changes, if a habitat changes, or if that individual moves to a new environment or habitat, natural selection may result in new adaptations. These new adaptations can sometimes give rise to an entirely new species. So here, for example, we have a population of, I guess they look like beetles. They have various inherited traits. Some of them make them quite brightly colored, others make them darkly colored. Now, this is a gene which controls the pigmentation of their shell. One sequence gives rise to a dark colored shell. A mutation in that same gene changes the biological metabolic pathway and leads to the production of a bright white pigment, so that mutation makes certain bugs brighter colored. That's not a big deal. I mean, both of these beetles are able to acquire food, both of them are able to exist in their environment, both of them are able to reproduce. But here's the problem. See that bird? That bird can see the brightly colored bugs better than the darkly colored bugs. And gradually over time, the birds start to eat the lighter colored bugs. That adaptation is not a helpful adaptation. It did not help the reproductive success of that particular beetle to be brightly colored. Eventually, over time, all of the brightly colored bugs are eaten by the birds, and the only thing you have left is the darker colored population. That is an example how a habitat or an environment can cause adaptations. These adaptations could be beneficial, i.e. giving rise to a dark colored beetle, or these adaptations could be harmful, giving rise to a lighter colored beetle. Natural selection has chosen for the dark colored beetle. The word science is derived from the Latin word scire, which means to know. And if you were to plug into the dictionary.com, what is science, or ask ChatGPT what science is, you get an answer like this. Science is the systematic study of the structure and behavior of the physical and natural world through observation, experimentation, and the testing of theories against the evidence obtained. This is a fancy way of saying we figure out how the physical and natural world works through a systematic study of what we see and how we explain what we see. At the heart of science is inquiry. In science, we search for information. We search for explanations as to why things do what they do and why things look what they look. And in the search for these answers, scientists use a process which is known as the scientific process. The scientific process includes making observations, forming a logical hypothesis, which is just a fancy way of saying I'm making a guess as to why something looks the way it looks. This hypothesis has to be testable, and you have to be able to prove it right and prove it wrong. So once you form that hypothesis, the scientific process continues on by saying, all right, you got a guess, now let's test it. Then you analyze your results from that test, and then you figure out, hey, was my guess right or was my guess wrong? So we verify the hypothesis. And that doesn't stop there. You don't just design one experiment and look at one set of results and say, hey, my hypothesis is true. You end up designing multiple experiments to prove a hypothesis. And once you've proven or disproven a hypothesis, the scientific process still doesn't stop. You keep making observations and you keep trying to figure out why things look the way they look, why things do the things they do. The experiments in this course, the laboratories that we are going to do, unfortunately, they are virtual, but I'm going to try my hardest to make them as close to what we would have done in our classroom as possible. I will show you videos. I have videotaped several of these laboratory protocols in my own garage, in something I call Biology 6 Garage Lab. And in these experiments, I will provide you with virtual data that you will then use to process. So the only thing you won't be doing in these laboratories is doing the lab, but you are going to process the data. Data is recorded observations or items of information. And the data that we generate in science is one of two things. It's either qualitative or quantitative. Qualitative data is descriptions rather than measurements. Quantitative data is recorded measurements. Now, one is not better than the other. For example, Jane Goodall, probably one of the most famous primatologists, the majority of her data has been qualitative, detailed, scrupulous observations and notes that she made of chimpanzee behavior. Myself, I am a molecular biologist, so a lot of what I did in my research lab at UCLA was quantitative data. I would put together things that I could measure, that I would then use and analyze and see if my hypotheses were right or wrong. Both of these types of data are utilized in experiments and are analyzed in order to prove a hypothesis right or wrong. They both have their place in science. As scientists, we collect data. And this collection of data can lead us to make conclusions. And these conclusions are based on a type of logic called inductive reasoning. Inductive reasoning allows us to look at data and draw a conclusion. It allows us to observe things and make a conclusion. These observations, these conclusions that we are coming up with, they often lead to what are called generalizations. So here's one. Bill and Tom are on the track team. I observe that. Bill and Tom are tall. I observe that as well. And I am going to, through inductive reasoning, make a generalization. I'm going to make a conclusion. Therefore, the track team members are tall. Well, that may not necessarily be true. Maybe it's just, I don't know, coincidence that all track team members are tall. But there's my generalization that I have derived through inductive reasoning from what I saw. The trick now is we've got to test that generalization and see if it's true. The generalizations that we make are really the starting point of what we call forming a theory. And a theory is a well-supported systematic explanation for an observed phenomenon.
But it's important to realize that these generalizations may not be correct. For example, being tall and being on a track team have nothing to do with each other. It's just a coincidence. So we have to test our generalizations through experimentation. We can't say that because penguins are black and white and old TV shows are black and white, penguins are old TV shows. You know, logic, something that a penguin's not very good at, but we as human beings are. And because we are logical, we can test these generalizations through some well thought out, well-controlled experiments. Observations and inductive reasoning can lead us to form a generalization that eventually becomes a well-supported, well-substantiated theory. But first, to test our generalization, we have to ask a question, and we have to propose a hypothetical explanation that we call a hypothesis. A hypothesis is a tentative answer to a well-framed question. I look at it as being a hypothesis as a really good guess as to why something is happening, why you are observing what you are observing. And you ask this hypothesis, you formulate this hypothesis before you even acquire data, because your hypothesis forms before you even perform the experiment.
Testing your hypotheses is going to require doing an experiment. Now, an experiment has to have two very important components. An experiment has to have a control. And a control is something that you can compare your experimental results to. There are two kinds of controls. There are positive controls and there are negative controls. The negative controls are going to show you what happens when your experiment doesn't work, and the positive controls are going to show you what happens when your experiment does work. The second thing you need for a good experimental test is repeatability. It isn't enough to do an experiment and get a certain set of data once. I want you to do an experiment and get the same or similar sets of data maybe 3 or 4 or 5 times. Is it reproducible? I cannot tell you the number of researchers I had who worked in my lab at UCLA who would come to me and say, "Professor, here are my results." And I would look at them and I would say, "Where's your controls?" I didn't have any. Well, then how do you know your experiment didn't work if you didn't have a negative control? Or how do you know your experiment did work if you don't have a positive control? And how many times did you do this experiment? Oh, great, it worked. It worked once. Great. Go and show me that it'll work 3 more times. And then maybe I'll believe you.
So let's come up with a simple example. You pick up a flashlight out of your drawer and you turn it on and it doesn't work. That's your observation. Your flashlight doesn't work. So of course, as a scientist, you're going to ask, what's going on? Well, first you're going to slap it in your hand and bang it on the table and turn it on and off 16 times. But eventually you're going to get to the point where you're going to say, why doesn't my flashlight work? Now, inductive reasoning could lead to a generalization like, my flashlight has a problem with something inside of it. Something inside of it's broken and it isn't working. And from this inductive reasoning, this observation, and this generalization, you can come up with 2 possible guesses as to why your flashlight has a problem. Hypothesis number 1 is the batteries inside of it are dead. Hypothesis number 2 is that the bulb inside of it is dead. It's burnt out. These are really well-framed guesses. They are logical, they are possible, and they are testable. Now, you'll notice that this generalization I'm saying of my flashlight has a problem with something inside of it leads to 2 really well-framed logical hypotheses. I'm not saying, oh, my flashlight's broken because the fairies came in and waved a magic wand and zapped it and made it non-functional. Okay, so, supernatural phenomena are not part of this. Your guess, your hypotheses are very, very well framed, logical, well thought out, and they can be easily tested.
Now, remember, we're going to need controls and we're going to need repeatability for a really good experiment. Let's concentrate on the controls aspect. It's something that you can compare, something known that you can compare your experimental results to. And we've all done this before. Your remote control batteries die, you go to your drawer and you see a bunch of batteries that are laying there and they've been there for God knows how long. And you pick them up and you put them in the remote and nothing happens. And you think, okay, great, my remote's broken. Or you go and you put some batteries in your flashlight and your flashlight doesn't work. And you think, oh, there's something wrong with my flashlight's bulb. Well, no. That may not be true. Just because one hypothesis may be proven false doesn't mean the other one's right. We'll come back to that. But you've done this before. Okay, well, wait a minute. Maybe the batteries I'm using in my flashlight or my remote control, maybe those are bad. So then you go hunt down something that has batteries you know that works, take them out, put them in your flashlight or your remote control. Did it work? Yes. Great, fantastic. It was the batteries. Again, just because you have eliminated one hypothesis does not mean the second one is the correct one. If you go and put in batteries that you know are going to work and your flashlight still doesn't work, okay, hypothesis 1 has been proven wrong. Does it mean it's the bulb that's not working? Nope. Now you have to go do a series of tests that prove it's the bulb. So we do this all the time in our everyday life. Change the light in your lamp. Okay, it didn't work. Oh my God, my lamp's broken. No, it's not. You just replaced the bulb with a bad bulb. Go and find a bulb that works and put it into your lamp. So hypothesis-based science most often has more than one hypothesis. And again, failure to prove one hypothesis doesn't prove the other ones right. If you replace your batteries and it still doesn't work, it doesn't mean it's the bulb. Maybe you're like me and you put the batteries in backwards. Inductive reasoning uses observations to make generalizations, and we use it to derive a hypothesis. Now, inductive reasoning can allow us to make stupid generalizations and bad hypotheses. Keep that in mind. But still, it's what we use to observe the world around us and make a pretty good guess as to what's going on. Deductive reasoning uses generalizations and a hypothesis to make specific observations in the form of a prediction. And my trick is deductive reasoning uses the phrase if and then. If all A are B and all B are C, then all A are C. That is deductive reasoning. Deductive reasoning is used after hypotheses have been developed, and it involves the use of if-then statements. If I put working batteries in my flashlight and it works, then it was the batteries that was the problem. Or if organisms are made of cells and humans are organisms, then humans are composed of cells. Inductive reasoning and deductive reasoning are sort of, two sides of the same coin, or they're sort of opposite to one another. Inductive reasoning is known as bottom-up reasoning, and deductive reasoning is often called top-down reasoning. So in inductive reasoning, we make an observation, and we see a pattern develop with this observation. And from this observation and from this pattern, we formulate a guess that we call a hypothesis. And through a bunch of testing, we eventually get to the point where we have a theory as to why the observation is being observed. Deductive reasoning, though, someone gives you a theory, and you now have to prove that theory. So you have to formulate a hypothesis, run an experiment, observe what you're seeing, analyze that, and then confirm whether or not the theory was right or wrong. So I guess in a way, inductive reasoning starts the whole process and deductive reasoning finishes it. Every scientific hypothesis has to be testable. It also has to be falsifiable. For example, a hypothesis that ghosts fooled with the flashlight cannot be tested. You cannot use supernatural and religious explanations. Those are outside the bounds of science. Science is a collaborative effort. It benefits when you have multiple people working in a team and they have diverse viewpoints. I always say that science cannot be done in a vacuum. I have a PhD in molecular and cell biology, and I started that journey back in 1992 when I started my master's and got my PhD in 1999. And all through that, I worked in a laboratory that was led by my mentor, and we had several people in that lab, and we often bounced ideas off of each other. I was one of her graduate students. Then I moved on to UCLA, where I was a postdoctoral researcher. And again, I worked in a laboratory that had numerous scientists, some graduate students, and undergraduate students that came in and out. And eventually I had my own lab at UCLA, where I had students coming in and out and where I collaborated with other people in the laboratory down the hallway, or maybe on another floor, or maybe even at a different campus. Because we talked to each other, because we communicated with one another, we were able to share our thoughts and our theories and our observations, and we could collaborate with one another and design fantastic experiments to prove theories. We also communicated with one another through seminars, through publications, through websites. Again, science is never done in a vacuum. You don't work alone. You ask people, you work with people, you talk to people, and from those interactions, great ideas come, great experiments come, and great science results. Scientists build on the work of others. When I was in graduate school, I didn't come up with my idea out of nowhere. I was working on a project that my mentor had started a long time ago. When I became a postdoc, it was the same thing. And when I ran my own research laboratory at UCLA, yeah, I looked at previous articles, I spoke with colleagues, and I built on their work. I myself published numerous papers that other people have cited in their work. So scientists check each other's claims by performing similar experiments. I know it seems like we don't trust each other, but we don't trust each other. So guess what? It's check and verify. It's not unusual for different scientists to work on the same research question. You know what? A little competition can be healthy, but also it's very, very helpful to have different scientists come together to collaborate on a similar project. And scientists can cooperate by sharing data with one another, by sharing their results and their conclusions through what is called the peer-reviewed publication process. We take our scientific work and we publish an article, and before it even gets published, it is submitted to peers in our field for them, quite frankly, to rip into a million pieces so that you can find the flaws in your work and improve them. When you see an article which has been published and peer-reviewed, you can pretty much all be guaranteed that it has been put through a rigorous critical process. And what you are seeing is well-researched, well-performed, and well-accepted data and conclusions. Now, this course will discuss what we know, using data that has been accumulated by many, many scientists, all working together over decades and building upon one another.